There is a cell in your body that will never be replaced, not once, for your entire life. And less than 3 feet away from it, inside that same body, there's another cell that will be completely swapped out for a new one before this week is over. Both of them rely on the exact same biological process to stay alive. That process is called autophagy, and the way it actually works inside each of these cells could not be more different. Autophagy means self-eating, and the mechanism behind it was mapped out in detail by Japanese biologist Yoshinori Ohsumi, work that eventually earned him a solo Nobel Prize in physiology or medicine. Stripped down to its basics, it's a recycling system. A cell identifies its own damaged parts, worn-out proteins, malfunctioning organelles, structures that have simply outlived their usefulness, wraps them up, breaks them down, and reuses the raw materials for something new. Every cell in your body runs some version of this. What almost nobody explains is that some version is doing a lot of heavy lifting in that sentence. The word autophagy hides just how differently this process behaves depending on which organ you're looking at. In the brain, it runs as a permanent, unforgiving operation with zero margin for failure. In the gut, it runs alongside a workforce that gets replaced on a rolling basis, which changes what the cleanup system is even responsible for in the first place. By the time you get to the 9th section of this video, you'll understand exactly why a single word in biology can describe two systems that barely resemble each other, and why that difference has consequences for two of the most common categories of chronic disease in the modern world. Two organs, same molecular toolkit, two completely different relationships with time. Here's the detail that changes everything about how autophagy has to work inside the brain. Neurons, for the overwhelming majority of your body's population of them, do not divide. Once a neuron finishes forming during development, it is, for almost all practical purposes, the exact same physical cell for the rest of your life. Skin cells slough off within weeks. Red blood cells get replaced on a schedule measured in months. Neurons, by contrast, are largely stuck with whatever damage accumulates inside them because there's no younger replacement cell waiting to take over. That fact was put to a direct experimental test in 2 separate research efforts, published around the same time, both in the journal Nature. One team, led by researcher Masaki Komatsu, engineered mice to lack a gene called ATGE7, a gene essential for building the autophagy machinery, specifically within the central nervous system. A second team, led by researcher Taichi Hara, ran a parallel experiment removing a related gene, ATG5, also restricted to neural tissue. In both cases, the mice developed abnormal limb-clasping reflexes and a visible reduction in coordinated movement. Komatsu's mice showed massive neuronal loss concentrated in the cerebral and cerebellar cortices, and none of them survived past 28 weeks of age. What made these 2 studies significant wasn't simply that autophagy mattered somewhere in the brain. That much was already assumed. It was that these mice carried no disease-associated mutant proteins whatsoever. Just removing the ordinary cleanup process, on its own, in an otherwise healthy genetic background, was enough to kill neurons and shorten the animals' lives dramatically. That told researchers something specific. Neuronal autophagy isn't a system you call in during an emergency. It's constitutive, meaning it runs continuously in the background because there is no other mechanism available to keep an irreplaceable cell functioning for decades. Now flip over to the gut, and the entire logic autophagy operates under changes. The epithelial cells lining your intestine are among the fastest renewing tissue anywhere in the human body. Stem cells sitting at the base of small pocket-like structures called intestinal crypts divide continuously, and the daughter cells get pushed upward and outward, replacing the entire functional lining of your gut within a matter of days rather than decades. That single fact changes what autophagy is even needed for in this tissue. One particular gut cell type, called a Paneth cell, sits near those crypt stem cells and produces antimicrobial peptides packed into secretory granules, essentially acting as a chemical gatekeeper that helps decide which bacteria are allowed to colonize the surrounding intestine. A study led by researcher Ken Cadwell, which examined both mouse models and intestinal tissue taken directly from human patients, found that a specific autophagy-related gene, Ag16L1, plays a direct structural role in how Paneth cells assemble and release those granules. When that gene was disrupted, granule formation broke down, and the same gene variant has been independently linked, in humans, to increased risk for Crohn's disease. One of the more than 30 genetic loci researchers have connected to that condition. So while the brain leans on autophagy to keep a single irreplaceable cell functioning across an entire lifespan, the gut leans on a version of the same machinery to manage an active, ongoing negotiation with trillions of bacteria inside cells that are only ever going to exist for a matter of days, regardless of what happens to them. Quick question before we keep going. Between what you just heard about the brain and what you just heard about the gut, which one caught you off guard more? Drop it in the comments. I actually read them, and they shape what gets covered in future videos. Something else rarely gets mentioned when people talk about autophagy casually. It isn't one single mechanism wearing different names. There are multiple distinct forms of it, and two of the most heavily studied are macroautophagy and chaperone-mediated autophagy, usually shortened to CMA. They don't just differ slightly. They work through entirely different physical processes. Macroautophagy is the bulk version, and it's the one most people picture when they hear the word autophagy at all. A double membrane forms around a chunk of cellular material, anything from a damaged organelle to a tangled cluster of misfolded proteins, seals itself into a bubble-like structure called an autophagosome and delivers the whole package to the lysosome to be broken down. This is the exact mechanism both the Komatsu and Hara mouse studies were built around, and it operates in essentially every cell type, brain and gut included. Chaperone-mediated autophagy works through a completely different physical route. Instead of scooping up bulk material indiscriminately, a chaperone protein identifies one individual target protein at a time based on a specific short sequence tag researchers call a K-FERC motif, then delivers that single protein to a receptor sitting on the lysosome surface, called LAMP2A, which threads it through one molecule at a time. This selective, one-at-a-time process was first characterized by researcher Ana Maria Cuervo, and it turns out to matter disproportionately for neurons specifically. Several of the proteins most strongly associated with neurodegenerative disease, including alpha-synuclein, are handled largely through this exact pathway. So what actually happens, physically, when a neuron's autophagy system fails? Returning to those same mouse experiments, when researchers removed the relevant autophagy genes, damaged proteins didn't simply build up in a diffuse random way. They clumped together into visible structures called inclusion bodies, composed largely of proteins tagged with a molecule called ubiquitin, marking them for disposal that never came. These clumps grew larger and more numerous the longer the affected mice survived. That experimental detail lines up with something researchers have observed separately in human tissue. Protein aggregates are a defining, consistent feature of neurodegenerative conditions, including Parkinson's disease and Alzheimer's disease. It's worth being precise about what kind of evidence each side represents, though. The mouse studies demonstrated, through direct genetic manipulation, that removing autophagy causes aggregate buildup and neuronal death in a controlled experiment. The human findings are observational. Aggregates are present in diseased brains, and notably, They also turn up in some aging brains that were never diagnosed with any disease at all. Either way, the underlying structural point holds. A neuron that fails to clear its own internal waste has no way to hand that problem off to a replacement cell, the way gut epithelium does routinely. Whatever accumulates stays inside that exact same cell for the rest of that cell's remaining lifetime, with nowhere else for the damage to go. Every square inch of your intestinal lining sits in direct, permanent contact with trillions of bacteria. The overwhelming majority are harmless, and a good number are actively beneficial, quietly helping digest what your own enzymes can't break down. But among that enormous population, a small subset are pathogens, and every so often, one of them manages to breach the epithelial barrier and get inside a gut cell. When that happens, autophagy stops behaving like a maintenance crew and starts behaving like a security response. This specific version of the process has its own name, xenophagy. The infected cell tags the invading bacterium, sometimes with a small protein called ubiquitin, sometimes with a sugar-binding molecule called galectin-8. And that tag recruits the same autophagosome-building machinery used everywhere else in the body, except this time the target is a living organism rather than a burned-out organelle. One of the more revealing examples involves Shigella flexneri, a bacterium responsible for severe intestinal infections worldwide. Research led by Michinaga Ogawa found that this bacterium had evolved a specific countermove, a protein called XB that blocks this specific defense at its source. Shigella's own surface protein, VIRG, normally attracts the host autophagy protein ATG5 and triggers exactly the kind of targeting just described. IceSB works by physically competing with ATG5 for that same binding site on VIRG, occupying it first and shutting the response down before it can start. It's a genuine molecular arms race happening inside your own intestinal wall, and most people go their entire lives without knowing it's taking place. That's a recognition problem neurons almost never have to solve. Under ordinary conditions, bacteria don't live inside brain tissue, so neuronal autophagy can stay entirely focused on internal housekeeping. Sorting damaged proteins, recycling old organelles, nothing more complicated than that. Gut autophagy, by contrast, spends a meaningful portion of its time making split-second judgment calls about what belongs inside the cell and what's a threat that needs to be captured and destroyed. It's a distinction that barely exists anywhere else in the human body. Here's a number that tends to catch people off guard the first time they hear it. The brain accounts for roughly 2% of your total body weight. Yet at rest, it consumes about 20% of the oxygen your entire body uses. That figure comes from a widely cited energy budget analysis by neuroscientists David Atwell and Simon Laughlin, and almost all of that oxygen is being spent powering mitochondria, the structures inside neurons responsible for generating the energy behind every electrical signal your brain sends. That level of energy production comes with a structural cost that plays out differently depending on the shape of the cell involved. Mitochondria degrade with use, and damaged ones need to be identified and broken down through a selective process called mitophagy. In a neuron, this isn't a tidy, localized task, because a single neuron can extend a long, thin projection called an axon far beyond the boundaries of its own cell body. And mitochondria sitting out along that length still need to be managed, monitored, and cleared when they fail, regardless of how far they are from where the cell's main machinery is concentrated. Gut epithelial cells never face anything close to this geometry problem. They're small, compact, and structurally simple compared to a neuron, and because they're replaced wholesale every few days anyway, there's rarely enough time for mitochondrial damage to accumulate into something autophagy even needs to address. The brain is running a decades-long maintenance operation inside oddly shaped, energy-hungry cells with an unusually large physical footprint. The gut, by comparison, barely has to think about it. When food stops coming in, autophagy doesn't switch on everywhere at the same moment or at the same intensity. One study, led by researcher Akiko Kuma, looked specifically at newborn mice suddenly cut off from their placental nutrient supply at the moment of birth, and found that autophagy activation didn't ramp up uniformly across tissues. Some organs responded faster and more intensely than others. That particular experiment wasn't about adult fasting, but it demonstrated a principle that carries over more broadly. Organs simply don't receive or respond to a nutrient scarcity signal in lockstep with one another. The brain faces an obstacle the gut doesn't have to deal with at all: the blood-brain barrier. This tightly regulated boundary controls, molecule by molecule, what's permitted to cross from circulating blood into brain tissue, which means the brain is largely shielded from the earliest chemical signs of fasting. Instead of reacting directly to falling blood sugar, it waits on the liver to begin producing ketone bodies from fat breakdown, then gradually shifts toward using those as an alternative fuel source once glucose availability drops low enough to matter. The gut has no such filter standing between it and the bloodstream. Its cells sit in direct contact with whatever is or isn't circulating, both in the blood supply and in the nutrients still moving through the intestinal lumen, so shifts in nutrient availability register there almost immediately. One organ finds out about fasting through a heavily guarded checkpoint. The other one finds out in real time, with nothing standing in the way. When autophagy fails in the brain, the result is degeneration, a slow compounding accumulation of damage inside cells that were never going to be replaced to begin with. The protein aggregates discussed earlier, including forms of alpha-synuclein, normally managed through chaperone-mediated autophagy, show up consistently in conditions like Parkinson's disease, where that specific selective pathway appears to lose function over time. When autophagy fails in the gut, the outcome looks nothing like that. Instead of permanent cell loss, the more common consequence is a breakdown in barrier integrity. The tight junctions holding neighboring intestinal cells together start to weaken. An inflammatory signaling pathway involving a protein complex called NF-κB becomes more active than it should be, and the tissue becomes increasingly prone to chronic inflammation. This is part of why the ATG16L1 gene discussed earlier, when its function is disrupted, has been linked specifically to Crohn's disease in human patients, not to any neurological condition at all. Line those two outcomes up side by side, and the same root cause, a loss of autophagy, produces two entirely different disease categories purely because of what kind of tissue it's happening in. A permanent cell breaks down irreversibly. A renewable one becomes chronically inflamed instead. Same failure, same underlying word. Opposite consequences depending entirely on where in the body it strikes. Autophagy was never one process wearing two different costumes. It's closer to a shared toolkit that evolution adapted separately for two very different problems. A cell that can never be replaced, existing right alongside cells that get rebuilt on a schedule measured in days rather than decades, in the same body, running side by side for an entire lifetime. That's the actual answer to the question this video opened with. The brain and the gut aren't running the same program at two different speeds. They're running two genuinely different programs that happen to share a name, shaped by two entirely different evolutionary pressures. Keep one irreplaceable cell functioning indefinitely, or manage a constantly renewing workforce under permanent bacterial siege. Both problems, solved with the same basic biological tool, pointed in two completely different directions. If this comparison changed how you think about that one word, autophagy, do three things before you go. Hit like, since it's the fastest way to tell YouTube this kind of video is worth showing to more people. Subscribe if you want more deep dives like this one, and send this to the one person you know who'd actually sit through a comparison between neurons and gut bacteria. See you in the next one.